the monitor on me. I don't want it following me. So good morning. This is how much I love you guys. I'm writing on boards, right? This is way out of my comfort zone, but y'all are worth it. So similar to, and I wanted to kind of start at the end, and although I'd like to start at the beginning of the story of immunology, right? Some of this stuff we didn't finish, and I want to go over it first, okay? So not exactly how I wanted to go, but we will start here. So, even got fun color coding. So, poor little antibodies, right? Which are also referred to as immunoglobulins, abbreviated IG, right? And there are actually 5 different classes. And I dug out my graduate level immunology book last night. Because, you know, when you don't cover the details for 10 plus years, right, you tend to forget the details that you guys don't need to know. But sometimes you guys ask me those questions, right? And then sometimes I even ask myself those questions and I couldn't remember them anymore. And looking at it, I was like, oh my God, yeah, I forgot. Like, there's not only 5 different classes. Within one of the classes, there's 4 different types of antibodies, right? The good news for you guys is we're going to stick to 4. And I actually even took a snapshot of the picture and put it in the PowerPoint for today. And there's a couple of slides from my graduate level textbook, which of course I'm not going to ask you guys specific questions on, but just so you could see, right? Some of this stuff. And I grabbed some pictures from another book too that had some really great tables and stuff that I think will help you with the content that's on the exam this week. So starting out with antibodies, we draw these poor little things as these little stick figures. If we were talking about molecular-wise, what type of molecule are they? They're a protein, right? They're proteins. Remember that, like, if you're not sure, when it comes to a molecule in the body, say protein, you're probably right. Because they're the workhorses, right? They do most of the work. They're your enzymes that catalyze reactions. In this case, it's a protein. And it's made of actually 2 identical protein chains, right? One of them is small, right? Denoted in purple here. And because of that, we call that one what? Not short, but light. Because it was smaller, it was lighter when they broke up this protein into the different pieces, it was the lighter one, right? So this is our light chain. But you produce 2, right, for an antibody. 1 for either side. Okay? And then 2, they're longer, so therefore they were heavier, so they're called the heavy chains. Let's see if I can write heavy and not mess it up. All right, hopefully that's right. Did I put my E in the right spot? Okay, so that's the general single monomer of an antibody, right? Your cells know how to make this because where is that information stored? In the DNA, right? And each B cell, question I wanted to ask you, but I just answered, makes their own unique antibody, right? So the answer here is this is a B cell, and it actually puts the antibody on its surface as its receptor, right? So B cell. Okay, so some more parts of an antibody though, right? So say this is some foreign substance, right? And we typically refer to those as antigens, right? Because they caused our bodies to generate antibodies. That's actually what it means. Antigen means antibody generators, okay? But we know that whatever this is, whether it be a foreign protein, a virus, right, a bacteria, it has smaller molecular components to it. That I denoted by this little red here. That's what can physically bind with the antibody, right? So what would we call that specific little piece of a larger antigen that actually binds to a receptor like an antibody. Epitope. That's the one I have right here, so I'll spell it right. Right? So, the good news, right, is for your test, you don't have to spell any of these words, you just have to pick them out, right? Oh, and I'm writing in the wrong spot. That's wonderful. See? Not well rehearsed. Ugh. Remember I said out of Erica's comfort zone? We're going to get to this piece next. But this piece, I'll write it in red. Or let's write it in black, because I'm not a big fan of red. But y'all understand. It's color-coded. For us that like color, but... Thank you. I was just cheap copy off paper. So that's the epitope, right? That's actually binding with, you see I have my antibody here. Okay. This portion of the antibody is really important to know the name of it because this is the part that the immune system is going to interact with. Right now it's embedded in the membrane of the B cell. But remember, B cells are going to turn into another cell type that's actually going to secrete the antibodies. And look, I got real lazy and drew it as an extreme stick figure. Right? But what do we call this cell? Plasma cell. That I think I can spell. Anyone know why we call it a plasma cell? Did I spell it right? Yeah, I tried to draw it as best as I could. It's got a lot of endoplasmic reticulum, right? So the plasma, the cytoplasm of these cells gets huge because its rough ER gets really huge. So not just ER, but RER, because what does that first R stand for? Ribosomes, right? Not only rough, but it's studded with ribosomes, which is why it appears rough under electron microscope. So if we, and it's a protein that's secreted, so the other thing involved would be the Golgi apparatus, right? So again, ribosomes make what? Proteins. What are antibodies? Proteins. Makes sense, right? Okay. So eventually, whatever antibodies on this surface, right, when it proliferates, differentiates into this plasma cell, right, it's going to secrete that exact same antibody. Each B cell is unique in that. But now these antibodies, right, are secreted. They can go different places in the body. I've got a cool picture for you guys for that later. Okay. Some of them stay as this nice, cute little monomer. Some of them form what we call a dimer. Actually, only one of them does this, right? Hence the blank. One class, right? And one of them, 5 of the monomers will get hooked together, right? And form this huge pentamer, right? And only one class does that, right? So we'll fill in the blanks in a second. But you'll notice, especially for this one, it makes sense that the immune system can access this, right? Because this portion is stuck to the antigen, right? So this portion is sticking out. What is this portion that's sticking out that's really important we need to label? Right? Some people call it the stem, but I want you guys to call it something else. The FC, right? F for fraction, C for the fraction that crystallized, right? But you'll see FC a lot because, of course, certain cells of the immune system have receptors for this, so they can grab onto this. Right? Because antibodies themselves, all they really are doing is helping us specifically, down to the molecular level, detect a foreign substance, right? Okay? Still with me? Okay, so let's go over the different classes. So the monomer one that is the most prevalent throughout your body, right? It'll be in your blood. It'll go into your tissues For women, we actually produce this in our colostrum and give it to our babies. It's one of the very first gifts we give them. IgG, right? And this is the one that also can cross the placenta, right, when we're pregnant. Right? So this is like one of the biggest ones. Okay? Another one that I hate, so usually my students remember this one, is involved in allergy. And it'll actually bind to mast cells, right, and basophils. And when the allergen attaches to them, those cells degranulate and release things like histamine. IgE, right? Not named after me, but I feel like it most days, right? IgE for Erica, right? This is allergy, right? Not so pretty one. And the other class that we didn't talk about is D, right? Would be in this monomer form, and we're not going to. But A can also be in its monomer form, but it can also form which one of these? The dimer or the pentamer? The dimer, right? And this dimer goes somewhere, right? And I always remember A for most abundant, right? And it goes somewhere. It actually does. It kind of stays in your body and kind of doesn't, depending on how you feel about the digestive tract. The tube goes straight through your body. It's actually considered an external environment, right? So what do you produce in that area in your respiratory tract? You produce a lot of what? Mucus, right? And some mucus has special names like tears and saliva and things like that. But all of that is bathed in mucus, right? Because that's like where stuff could potentially enter into our body, right? We can inhale it in, we can ingest it in, right? So we want antibodies right there to be able to bind to it. But remember this is outside of your body. So are there phagocytic cells there or T cells or anything? No. So how could those be protective? We're going to talk about it. Right? So that's A. What are we left with? M, right? And what was one of the clues that I helped you remember this one? It's mega big, right? I always remember M for mega big, right? So this one's huge. It can bind up a lot of antigens, right? So antigen binding sites wise, 2 for just the standard monomers, right? 4 here. So therefore, how many here? 10. 10 antigen binding sites. And there was one picture I didn't get to last time that I want to show you, and I will today. All right. So that's general stuff about antibodies, right? Definitely could be a really good matching question, right? On the test. Is a matching question on the test. All right. So, and then look, I drew this yesterday. So I said, please. I cleaned all 6 of these boards yesterday too, because they were not as pretty as they are right now. All right. So, and I got my list. Right? So, okay, great. Antibodies bind antigen. So what? Why is that beneficial? These 6 reasons are why it's beneficial, because these are the 6 possible ways in which the immune system is going to respond when an antibody is bound to an antigen, specifically at its epitope. Right? So I have some pre-drawn things, right? And one I haven't drawn yet. So take a look at it for a moment. I'll try and step out of the way. I'm recording, remember. Okay, so what do you think? Which one did I not draw? 1, 2, 3, 4, 5, or 6? Now I got that one. It's hard to differentiate because it's not labeled real well, but it's there. All right. I'm going to start drawing. See if you guys get clued in. All right, so what'd I go with? Red was the bad guy. That's appropriate, right? So, draw me some bad guys. Some antigen, some foreign protein, bacteria, virus, doesn't matter what it is. And blue was my antibodies, right? Let's draw me some antibodies. Agglutination, right? So just because at the very least, right, an antibody has 2 antigen binding sites, right? I drew only a little monomer, right? But that monomer has 2 antigen binding sites, so it can hold on to 2 antigens at the same time. And when you have more when you have the right ratio, you get this clustering effect. Well, why would that be beneficial? It can't go anywhere, right? So especially if it's like, say, a motile bacteria, we can immobilize it, right? We can get it stuck there. So again, IgA as a dimer too, right? So now we're talking about something that looks like this and can do more binding, right? You actually secrete into your mucus, so you can clump up the bacteria like this. Or like he said, you know, if it's a motile bacteria, we can stop it from moving, right? Think about viruses. What's the first thing they have to do? Attach to us, right? If we're clumping them up, it's going to make it harder for them to do that too, right? And has an additional protection because it's stopping it from binding to us, which we consider a separate outcome on here. But this is agglutination. So this is 4. I won't worry about color for now. So some antibodies are better at this than others. So obviously IgA is really good at it because of the 4 antigen binding sites. Who would be the best at it? IgM, because he's so big, those 10, right? So I've got a picture for you guys later. I'll show you showing that, because I'm not going to draw that out because that's not easy to draw. Right. Not in a short time frame either, right? So, okay. The next one, right, I'll start telling you the story. I'm going to work on this one. So here's this ugly worm, right, and it's got things that an antibody can recognize. And there's a particular cell that has a receptor for this portion of the antibody, which is what portion of the antibody, though? This is the FC portion. So this is an FC receptor, right? Okay, everyone agree with me? But it is for a particular class of antibody that some of you guys started to say. Can't squeeze it in there, good. So I'll give you a clue, right? This guy's granules, right? He's one of the fills, right? And is releasing granules that contain things like histamine, right? Basophil or a mast cell, right? So this would be IgE, right? This would be IgE. So again, just like we talked about before, this one would be 5, but I'm just showing you basophils and mast cells here, right? So here would go basophils. Oops, no. What is that? That's a granule, right? Or a vesicle, right? But this cell itself is a basophil or a mast cell, right? Eosinophils, right? They have granules too, and they can release stuff, but different stuff from basophils and mast cells, right? And that's because they can't grab onto this thing. He's too big, right? They can't take it in and digest it. But some of our cells can do that, right? We do have phagocytic cells that if it's smaller, like say a bacteria, right, and an antibody is bound to it, and again, what is this receptor right here? That's an FC receptor. So this is a phagocytic cell, right? We don't call these, they're vesicles, right? But we don't call them granules because our phagocytic cells are who? They're neutrophils, right? Who else? Yeah, dendritic cells you can say do phagocytosis. That's not their main job, right? How do these caps fit on this? Okay, there we go. All right. Named after the fact that the nucleus tends to be mono like this. Monocytes, right? Which are called what in our tissues? Macrophages, right? So your main phagocytic cells are going to have this FC receptor, right? Are your neutrophils, your monocytes, and your macrophages. I didn't leave room for writing on here. Y'all can figure it out. This, on the other hand, right, because it's part of phagocytosis that we're going to look at later, is called what kind of vesicle? It's named after an enzyme it typically has in here that, especially if this is a bacteria, he's in trouble. Lysosomes, right? Because lysozyme, which is the enzyme, is usually there, right? So that's another one I will misspell all the time. Go ahead, ask your question. I'm not going to ask you specifics, but okay, so he asked about hydrogen peroxide, right? And nitric acid. What do all of those have in common? They destroy bacteria how? What do they all what element do they all have in them? Oxygen. So that whole process is referred to as what? What do you take in when you respire? Oxygen, right? It's called respiratory burst, right? So if it's got oxygen in it, right, it's involved in respiratory burst. Yeah, I'm not going to expect you guys to list all those on that list. I don't even have that list memorized. All right, but I wanted to write down lysosome, right, before I forget and don't write it. I have a good speller in the class. I get all messed up in the SOMs. I have horrible handwriting. Okay, lysosome. So which one is this, though, on our list? Really? All right, we already did 4 and 5. I didn't put my 5 in here. Let me put my 5 in there. We did 4, right? That's opsonization, right? Tagging for destruction, right? Whenever, and this is with the antibodies, but we learn about other stuff in our innate immune response, right? They can tag for destruction. Phagocytes have receptors for more than just antibody, right? They have receptors for C3b from the complement system, right? We'll hopefully get to that, right? So that one is 3, opsonization, tagging for destruction. That's how I always remember that one. Okay? All right. So, as we said earlier, one of the great things of binding them up like this, also called cross-linking or agglutination, or even precipitation if it's happening outside the body, right? We can make proteins precipitate out of a solution by adding antibodies to them. It's really cool stuff. Okay? So, by maybe stopping them from moving, right, clumping them up, okay? And additionally, by that antibody binding to, say, a toxin, a virus, or a bacteria. I started to write something there. I got interrupted. Oh, protein. Or a protein. For all these things, right, definitely toxins, definitely viruses, and sometimes bacteria and different proteins, they have to physically bind to our cells first, right, in order, one, for viruses to gain entry. Same thing with toxins. A lot of them, they'll bind to us and then they get taken into our cells. Some bacteria, too, are intercellular, right? So they bind to us and then go inside. Even the ones that don't go inside, a lot of times they still stick to us, so they can stay put in the body, right? So if we coat them with antibodies, we essentially stop them from getting in. This is generally referred to as, what do we got left? Neutralization. This is neutralization. This is number 1 on my list, right? These are just numbered for list reasons. Not for any other reason. Okay, so you seem to like this one, Colin. So we'll jump to this one, right? So antibody-dependent cell-mediated cytotoxicity, which means we're going to kill a cell, right? Cytotoxic means we're going to kill a cell using a cell. So which drawing looks like a cell killing a cell? Yeah, this one right here, right? But who is this cell? With this, yet again, FC receptor, right? And is going to kill, right? Say this is, for example, a skin cell, right? Could have been infected with a virus, and now we got viral proteins on its surface, or infected by a bacteria, right? But somehow, some antibody, and there's only one class of antibody that interacts with this one cell type. That's a pretty good killer, hence its name. That's the natural killer, right? This is the only one that can do this, right? So this is a lot of times abbreviated just NK cells. What antibody class? IgG. And it makes sense because remember we said, right, this antibody can pretty much go everywhere. Inside the body anyways. And just about any of your antibodies could do this, right? Any of them can do agglutination. They can do neutralization, right? Some are better than others at this, right? At opsonization, tagging for destruction. And some are better at this as well, right? And so the antibody binds, and then a protein that circulates in our blood binds. We're only left with one, right? Because we've already covered 6. So there's a clue here. What's that protein? It's a complement protein, right? So remember complement? This can start a cascade of events to happen, right? Once activated. And this is the what pathway when it's an antibody binding to a complement protein of complement activation. It's the first one we discovered. Classical, right? IgG is really good at this. IgM is really good at this too. Okay, so a cascade of events is going to happen, right? C1 is binding here, right? That's complement protein number 1. And then C3 convertase forms, and you get C3A and C3B. Oh gosh, you remember that? And then eventually, right, we can produce this structure that will literally form a donut-shaped protein structure, right, creating little pores in the cell, and it will cause that cell to lyse. So we could directly kill the cell, right, with proteins, but only because the antibody recognized it as being bad, right? So this is tying in 2 systems. Innate system with complement. Complement is innate, right? You're born with it. But antibodies is adaptive, right? Something that you make later. So what is this called, though, that donut-shaped protein structure? Membrane attack complex. Remember, that's important, right? It's commonly abbreviated MAC, but that M is real important, right? Because it inserts into membranes. So which bacteria is more susceptible to this, gram-positive or gram-negative, and why? Gram-negative because of their outer membrane. Gram-positive, remember, very thick layer, peptidoglycan. Those proteins would have to go through that layer to get to the membrane. It's harder. It can happen. But Gram-negative have that outer membrane. It's a membrane. These proteins can insert into it and lyse those cells. Okay? So these are all the myriad of possibilities that could happen when an antibody binds to an antigen, right? So we call these outcomes. Pretty awesome, right? See why they're my favorite protein, right? They can do some pretty amazing things. Okay, so as I run into the other whiteboard, there we go. I added at the end of the module this PowerPoint, right, as a review of some of the important stuff. So the cells, right, they all are born where? In our primary lymphoid organ of the bone marrow, right? Some of them, though, must travel to this guy, the thymus, though, to get further educated, get all their receptors right, all their markers on their surface. What cell type is that? T cells, right? They're named after the thymus, right? And then all of these lymphatic vessels in green, right, drain our tissue whenever we have inflammation or any other time. And along the way, they're going to filter that fluid that they picked up that we call the lymph, hence the name, through lymph nodes. Inside these lymph nodes is where, after they're born, our lymphocytes, which are B cells and T cells, are going to go to live, right? And they scatter throughout the body, too. And they're going to be those tonsils that are right in your throat. But remember, you have a tonsil in the back of your nose, right? The adenoid, not shown here. You apparently have one associated with your tongue. I've never heard of it. It's apparently quite lazy. I've never heard of that ever getting inflamed. So our secondary, right? So this is their home, right? Our tonsils, our adenoids, even your appendix is at that end of the hanging off the cecum, just before the large intestine. Chock full. That's why sometimes that can get inflamed, right? Stuff gets stuck in there. You got a lot of white blood cells. It gets inflamed. The bad thing is if that bursts, that's a hole in your intestines leaking into the inside of your body. Not cool. Your spleen, right there, helps capture pathogens found where? In your blood, right? In your blood. And it's the only organ they're not going to replace if you lose or it ruptures or something bad happens to it. But it leaves you more susceptible to bloodborne pathogens, right? Because you don't have that level of protection anymore. So, in the bone marrow, as it says here, right, we have progenitor cells that give rise to all the blood cells, right? So even the red blood cells, the megakaryocytes that break up into platelets, which we didn't talk about, right, but briefly, the fact that they can wall off things. They're like little bags of enzymes. They're pretty amazing. Mast cells, which, typically you're going to find in your tissues, and they're a close cousin to the basophil. Notice the coloring is very similar. Then you have your lovely granulocytes. All their names end in "phil." They're all named after how their vesicles stain neutrally, basic, and with eosin, which is an acidic dye, this reddish color. Monocytes can give rise to macrophages in our tissues, right? And dendritic cells. And again, dendritic cells are named after the fact that they have those long cytoplasmic projections because they're the really important ones for antigen presentation, which I definitely want to talk about. Natural killer cells, which are lumped in with lymphocytes a lot of times. But these guys don't recognize antigen on their own, right? They needed the antibody in the case of antibody-dependent cell-mediated cytotoxicity. They'll kill cells that aren't presenting MHC class 1, right? Because remember, that's like our ID card. If you're not presenting that, we're going to kill you. Some viruses downregulate that because, of course, they don't want to be found out. They're like, don't present my information on the surface. Please, don't do that. And then we have T cells, which we know there are several different types of T cells, right? And even subsets of that. And then B cells that can differentiate into just plasma cells, which are going to secrete antibody. So this is a good pictorial representation. This is a really good one as far as functionality goes, right? Our neutrophils, which are the most abundant, right, are really good phagocytic killing cells, right? Eosinophils, these are the ones involved. Their granules contain things that are important for inflammation and allergy. So are basophils and eosinophils, right? These guys kind of counteract each other. It's the basophils and mast cells that release histamine, right? Monocytes, macrophages, and dendritic cells, again, fall into this mononuclear phagocytic cells, right? So they all have the ability to phagocytize, but notice again, this one really talks about the next step of presenting that information. And in lymphocytes, right, T cells, B cells, natural killer cells, but mostly the B and T cells are really when we get into adaptive immune. So how do we know the bad guys are there? All our cells, and more so on cells that are like phagocytic cells that are supposed to recognize these things, have receptors on their surface, right? And this is part of our innate system, right? We're born with this, that we can recognize things have gotten in, signal gets sent to the command center, right? Cytokines are released, or a specific cytokine for a viral infection known as interferon. Right? So remember interferon. So again, interferon is what system? Innate, right? Adaptive is what vertebrates have, right? That's the add-on, right? That's the specific recognition with antibodies and B cells and T cells and all that cool stuff. What's the special thing about apoptosis? Programmed cell death without inflammation, right? But we're going to stop the cycle of a virus, hopefully, with that. Cytokines, remember, are our chemical messages, right? So IL is interleukins. These are white blood cells talking to each other. TNF is tumor necrosis factor. In general, right, this cytokines themselves, right, I don't want you memorizing a whole bunch of them. There was only one I said that was important. IL-2, right? And that is real important for T cell proliferation. Both T cells. Okay? So how do phagocytic cells get activated? Again, they have those pattern recognition receptors, right? All these guys, right? These are examples of the toll system, toll-like receptors. But they recognize patterns, right? This doesn't tell you that this is, staph, Coccus aureus, right? It says, no, it's a gram-positive bacteria, right? LPS, right, tells you it's a gram-negative. It doesn't say it's E. coli. But, hey, we don't really care. We just know, hey, it's a bacteria. It's not where it's supposed to be. We need to respond accordingly, usually inflammation, right? Or if it's a virus that got in, right, we may want interferon to shut down that replication cycle. Other receptors, remember, see, very important, FC receptor, right, for antibodies. But, IgG is the big one in the body. But we can recognize other things, right? Complement, C3b, right, from the complement system. That's an alternative pathway is always producing this. If it binds to a bacterial cell, this is going to help a phagocytic cell recognize it's a bad guy. Mannose binding lectin, C-reactive protein. These all bind to, these are proteins that we produce and bind to the bad guys to help us recognize them. Other than antibody, that's all innate. That's it becomes built in. Antibodies, right, are adaptive immune response. For some things I do, yes. Yeah, I have some pictures in this one. I'll tell you all that. Help you out, right? So although pattern recognition receptors, right, and cytokine signaling, right, effective killing machines. So a lot needs to happen, right? So not just these receptors, right? Remember, and probably, as I'll show you in a moment, right, we're just scratching the surface when it comes to this class, right, of the interactions that happen. So this is an example, right? So we have choices, things that can happen, right? We just saw mannose binding lectin. That protein binds to mannose, a protein found on bacterial cells, right? It can activate complement via the mannose binding lectin pathway, the third pathway for complement activation. And we can get that MAC complex and we can destroy those cells. Or another way they could be destroyed is a phagocytic cell, which has a receptor for it, engulfs it, and destroys it by phagocytosis. That's innate, y'all. We could kill the bad guy before we even make antibodies, right? That could happen, right? So this is a good summary slide of, right, complement. Classical pathway, antibody started it. Lectin binding, right, is mannose binding lectin starting it. And C3b, which is a component of the complement system, is the alternative pathway. Which is probably, these two are probably what happens first, right? This is innate. This is built in. This is if we made antibodies against it, right? Which we know can take about how long? 7 to 14 days, right? Once this system starts, it's like a domino effect, right? And there are major things that always happen. You get those C3 convertase and C5. So which one is the opsonin? Tagging for destruction. So C3b not only can start this cascade, right, but can tag for destruction. Notice, again, C3, where is it? It's not on here. Yeah, right there. Complement protein factor C3b. Pink one. We're going to get some that are going to help with the inflammation, right? Call more cells to the area, right? And we're going to get that MAC complex. These are all possibilities. Does that always happen? No, right? But these are the possibilities. These are the possible outcomes that can happen once complement is activated. This is innate, but, right, adaptive can turn on this innate system. So once we can recognize, we can bind to them, right? Some receptor on a phagocytic cell, right, is going to engulf it. What would we call that vesicle that we just engulfed it in? So this process is called phagocytosis, right? So that is called a phagosome, right? And these are called what? Lysosomes, because they tend to have what enzyme in high concentration? Lysozyme. But they could have any of those things part of the respiratory burst, right? They can make hydrogen peroxide, all those other oxides, all the ones that have oxygen, hence respiratory burst. And it can fuse with the phagosome, with the lysosome. So guess what we do with the name? We fuse it together, so now it becomes a phagolysosome, right? And we destroy this guy. And then they're going to spit it out by exocytosis. But if you are a dendritic cell or a macrophage, you're going to do more, right? You're going to take pieces and you're going to present those pieces. So there's with all the answers filled in, right? I remember all those lovely receptors and who has them, right? These are really important to know. And the most important is that ID card, right? MHC class 1. All our cells have it. All our nucleated cells, right? It's like the ID card. It's where stuff gets in, or even our own things. We're always presenting MHC class 1, right? Only a small group of cells known as antigen-presenting cells. You can even say the professional ones have MHC class 2. There's only 2 classes. And these are your B cells, right? And as you can see, macrophages and dendritic cells, right? Antigen-presenting cells. Because they need to present antigen. Who do they present it to, though? That helps in their activation. T cells, right? And T cells interact with both MHC class 1 or 2, depending on which T cell it is, right? B cells can directly react with antigen because on their surface they have that antibody, right? Which in this case, because it's embedded in the membrane, it's called the B cell receptor. T cells are going to recognize an antigen presented to them via MHC class 1 or 2, via their T cell receptor. But remember, there's a special handshake, right? We have 2 major groups of T cells, right? And they have a co-receptor that allows them to interact with either MHC class 1 or 2, right? So you have your 2 choices are CD4 and 8, right? 4 for helper T cells, CD8 for cytotoxic T cells, which says a lot about their names, right? Helper T cells do a lot of what? Talking, releasing cytokines, telling everybody else what to do. What do cytotoxic T cells do, on the other hand? They kill things. Cytotoxic, right? They will kill, right? Like the letter 8, right? 8, eat, right? Okay, but which matches with which? So CD4 helper T cells talk to MHC class 1 or 2? 2. CD8 with MHC class 1. Because again, think about this. This is our first line of defense. All cells present MHC class 1. If something gets inside of them, you want a cytotoxic T cell to be able to come by and recognize that and destroy that cell. Right? So notice neutrophils, FC receptor, macrophages, FC receptor, right? This is for that portion of the antibody that sticks out when they're binding to an antigen. Okay, so natural killer cells, right? Useful eliminating pathogens, right? When our cells, but again, not phagocytic, right? They release their granules, right? They're going to cause programmed cell death with granzymes, or perforin, where they're literally going to perforate the membrane. And even abnormal, our own cells, when they're not presenting like they're supposed to. So one big note, right, that I would definitely put there is about the MHC class 1, right? Cells that aren't presenting MHC class 1, natural killer cells are going to kill them. So that's not normal for them. And then we saw antibody-dependent cellular cytotoxicity, right? That's possible because of natural killer cells and antibodies. So B cell, right, because we have antibody on its surface, can directly interact with antigen. T cell, because it has a T cell receptor and a co-receptor, and it's having antigen presented to it by a cell, right? So these are 2 good images I clipped out from someplace else, right? It kind of puts these 2 side by side, our 2 major lymphocytes. So there's the dreaded word epitope, right? That's the little tiny piece that either an antibody in this case or a T cell receptor is binding to. But that T cell receptor is binding to it because some cell is presenting it via MHC, whether it be 1 or 2. So this is, again, a flow that could potentially happen, right? When we start at the very beginning, right? And the good news is you guys don't have to know which one goes cell-mediated versus humoral, but you do need to know that the 2 exist, right? Humoral is the antibody way, cell-mediated is a cytotoxic T cell. And we can go either way or both ways depending on, again, what signaling happens, cytokines, right? What communication, what receptors are tagged. So this is, again, another one I clipped out from another book because it is a much better table listing the properties and functions of the different antibodies. And it even has D on here, which you don't have to worry about, of course. There's that picture that I didn't want to draw. So as you can see, right, IgM, really good, right, because it's so big at agglutination, right, clumping stuff up, especially red blood cells. And so if you ever take Anatomy and Physiology 2 lab, this is the antibody you make against the ABO blood group. And so you get that really good clumping happening, right? And they'll literally take anti-A antibodies and put it in a well and add your red blood cells to it. And if this clumping happens, you know you have that antigen, right? They put anti-B. If it clumps up, you know you have that antigen. My blood is boring. No clumping happens. I don't have A or B. I am O, right? No clumping happens. I am Rh positive, on the other hand, so when you put that antibody, but that antibody we make is IgG. So remember, it's a monomer. That's why it's a pain in the ass when it comes to newborns, right? Because remember, that's the one that can cross the placenta. And that's why these, right, these are FYI slides, right? No specific questions from these slides on the exam. But I really liked this picture from my book because it shows you the distribution. And I think for some of us, like that picture in our mind of where these antibodies are. So IgE at the surfaces, right? So notice the pink line. IgG all throughout the body and crosses the placenta to the baby. IgA in its secretory form in that dimer, right, helps protect all of this, right, respiratory, digestive tract. We even produce it in our milk to give to our babies because, again, it will coat their respiratory and digestive tract. Mainly, of course, their digestive tract because they're drinking in the milk. When they're first born, too, they have receptors specifically to be able to take in the IgG that we produce in our colostrum. The heart is colored in IgM and IgG only to represent, and this is why descriptions are important to figures, right? To represent that it throughout your blood, right? So throughout your blood, that's pretty much mainly where IgM stays, right? And then, as I said, like we produce it in our breast milk. But this is the chart where I was like, oh yeah, that's right. So although like any one of them can neutralize, right? Who's the best at it? IgG, the best at neutralization, and IgA. So notice the pluses versus the pluses, right? But notice IgG 1, 2, 3, and 4, right? And I panicked when I saw this number at first, and then I was like, oh yeah, there's a minus sign right here. Very, very small quantities. Unless, of course, you're looking at my blood, right? And in that case, depending on what allergen you're talking about, I have lots of IgEs. But for the most part, it binds to those cells so you don't see it in blood. But serum levels, right? That's blood levels. So remember we talked about hemagglutinin way back with viruses, and influenza virus. You know, who better to talk about right now is influenza during flu season, right? So it binds to, right, the terminal celiac acid residues on carbohydrates of glycoproteins. Oh my God, right? What's the important thing here? Of epithelial cells of the respiratory tract. That's why, again, you inhale this in, right? And these are the major cells that are infected right away and why you feel so miserable. Okay? Where does it get its name? From the fact that it can clump up hemagglutinin, chicken red blood cells. So for some reason, right, they exposed chicken red blood cells to this and it caused them to clump, right? And so that's how it got its name. Toxins, right? A lot of bacteria produce a lot of toxins, right? And this is just a like small list. And the bad thing about these things is they can get into the bloodstream, they can spread real quickly, and as little as one toxin can destroy one cell. And I always get asked this a lot of times, like, why is anthrax so bad? Because toxin increases vascular permeability, leading to edema, right? So it basically causes inflammation, but you're not causing it; it's causing it, right? And so then you get you get hemorrhage of your blood vessels, and circulatory collapse. So this is why sometimes even we know someone's been exposed, right? And we go to try and treat them, it's just not fast enough. Because, I mean, that's probably the most evilest thing you could do to somebody, right? Is destroy their circulatory system. We're so dependent on that system for life. And so, yeah, you can, again, that's just FYI, right? Scare the shit out of you, right? Why to stay away from bacteria, especially toxin-producing bacteria? Those are the bane of our existence as we know it. All right, so let's think about this. Something gets into your body, right? What cell here do you hope grabs that if you can't destroy it by complement, right? Your phagocytic cells aren't recognizing it, right? Could be a virus, could be a bacteria, could be a foreign protein, right? You hope this guy is a dendritic cell, right? Because what system do we now need to turn on because either the first system's not taking care of the problem, right, or it needs help? Adaptive. Because innate, right, isn't taking care of the problem, right? You've got pus, right? You've got a wound, you've got pus. What does that say? Neutrophils are failing, right? You've got dead neutrophils there. You've got probably bacteria there, right? You're going to the doctor, right? Okay. But, right, one of our systems, right, that's going to get activated, and the way that it's going to get activated is this dendritic cell, right? This is the really important guy, is your dendritic cell. And he can do what's called cross-presentation, which I told you guys didn't have to worry about. And in essence, what it means is that he can present both ways, right? He can take stuff in, right, and present it via MHC class 1 and 2, right? He can do both. So can macrophages. Macrophages are a little bit restrictive in that, right? But if this is the very first time we've seen this bad guy, we haven't been vaccinated, right? The story has just begun, right? And we're not doing so great on the innate side. So this guy goes around, he gobbles up, right, and is presenting pieces of the bad guy, the red stuff, via MHC 1 or 2, or both, right? He can be doing both. But this is restricted. We already went over this, right? So pick a T cell helper, okay? So this is a T helper cell. So therefore it's CD4, which is this guy right here. This is the T cell receptor that's actually recognizing the antigen, right? So if we decided, right, we need help, we need the helper T cells activated, right? This would be MHC what? 2. Good. You guys are getting it. So if we're going this route, right, this guy can talk to other cells, right? What cells could he talk to? He can talk to B cells, macrophages, right? Big time talker, right? Okay. But in order to go B cell route, which is going to generate what? Plasma cells, which secrete antibodies, right? If we don't want to be stuck with just IgM, because remember, IgM can be produced without T cell help, right? That's a T-independent antibody. It's not a very high affinity, doesn't bind real well. That's okay sometimes, right? But sometimes we're going to need this T cell to help us go in that direction, right? If we want to go humoral, right, or antibody route. So in that case, we're going to talk to a B cell. Now remember, the B cell's got its antibody on its surface, right? But it can take it in and present the antigen via MHC too. Because again, this restriction of this pathway, right? This guy proliferates, right? Some of them become what? That's going to help us out in case the bad guy comes back again. Memory cells, right? So we're going to get memory. They'll hang out in that secondary lymphoid tissues, right? In case the bad guy comes back. Or the other choice, right, which we definitely want to happen, is big old plasma cells actually secreting antibodies of whatever class we need, right? So how does that plasma cell, it's really going to depend on where it is in the body, right? It's going to get that different signaling, whether it's going to do IgA as its dimer, right? If it's going to do IgM, the big huge one, obviously not going to draw that. Takes too long, right? So that's humoral or antibody route, right? We can go that way. But we also need these guys sometimes to talk to this other choice. So if this is a helper T cell, we would call this one a cytotoxic T cell, and this would be CD8. And this would be MHC1. If you can do like I'm doing right now, I'm going to ace this test. Okay. All right. So these guys, right, with the help sometimes of helper T cells, right, with cytokines, right, their talking ability, are going to produce these effector cells, right? What's one of the awesome things too? Some of them are going to be memory, which I can't spell apparently. That's okay. Close enough. Right? And then cells that are what we call effector cells, right? So they have that T cell receptor, and any cell, say a skin cell, for example, expressing, right, via MHC class 1, that antigen from way back here, they, on the other hand, are going to release stuff, right, like natural killers do, granzymes, perforin, and they're going to kill that cell. That pathway is referred to as cell-mediated, right? Because again, it's cell on cell. Make sense? Where humoral, right, is also sometimes often referred to as just antibody response. Right? Can we do both of these? Absolutely, right? So let's think about a virus. Which response do you want and why? You've got influenza. You want a humoral response, you want antibodies, why? So the antibodies will bind to them and do what outcome we talked about? Neutralize, right? Or we could get opsonization, right? We can clump them up, right, in the mucus, right? Because you breathe it in, right? So we get IgA produced, clump them up, push them out of the way. Don't even let it attach to our cells, right? Let's neutralize them. Or if they get in, we phagocytose them, right? Is complement activation really going to help us out there? No, right? Not that I'm aware of, right? Natural killer cells. Yeah, think about this. If a cell gets infected with a virus, it could be expressing viral proteins. If you have an antibody against it, right? Antibody binds, right? So antibody sounds awesome, right? For when the virus is where? Outside of your cells. But does it stay there? No, its goal is to get where? In your cells. Because that's where it's going to use your cell's machinery, your cell's ribosomes to make its proteins, right? All your resources and make more viruses. So if you have a cell infected with a virus, do you want to kill that cell? Yeah. Do you need to turn on cell-mediated? Yeah. Actually, with a virus, you want both, right? You want antibodies because, as we said, they can do all those awesome things, right? But you also want a cell-mediated response because they're inside your cells too, right? And then we have interferon that could be triggered even, right? That could help out. That's innate, right? Innately built in. Cells recognize. They warn the neighboring cells with that cytokine. They get set up and get ready. You don't even need adaptive, right? So why do we survive viruses? Because we have like all these ways to protect ourselves, right? And natural killer cells too, if they downregulate MHC production, because remember that T cell is only going to kill it if it recognizes what it's being presented via MHC, right? Natural killer cell, if it's not even presenting at all, is going to come along, ID card, please, right? And the cell's like, oh, no, I don't want you to know. And he's going to be like, well, too bad, you're done for, right? You're not behaving properly. I'm going to kill you. So lots of defenses, but we still get sick, right? Because at a certain point, right, it's more than the system could handle, or they downregulate these things, right? Interferon doesn't work, right? We have to wait for adaptive to kick in, right? We have very few antivirals. We've got, along with flu season this year, we've got the respiratory virus. It really is like we don't take care of the problem until adaptive kicks in. But for the most part, we're just trying to keep you alive, right? Until your immune system takes care of that nasty virus. What's the good news when it comes to bacteria? Kind of help out the system. We do have antibiotics, right? Stolenectology we can use. But yeah, sometimes you have to go all the way, right, to this level to get rid of a virus. But we can do it a lot of the time, thank goodness. Make sense? It's a lot, I know. And again, as you saw from my graduate level textbook, right? Just skipping the service. Just a summary of the awesomeness